ready to deploy. I am Rashmeet Sangari. I'm founder and CEO of SkyPreme. I have spent 20 years across global operations at J&J, Biogen, Amgen, and I have now built SkyPreme, which helps organizations get AI ready right from the start. Today, I'm very pleased to welcome Tang, Director of Bioinformatics at AstraZeneca. He went from working at a wet lab to leading cancer drug discovery through MD Anderson, Harvard, and Dana-Farber. He's built a following of 120,000 followers on LinkedIn and has a mission to reach 1 million people to teach computational biology. So please help me in welcoming Tommy. Great to have you here.
Thank you very much, Rashmit. Thanks for the invitation. Yeah, as Rashmit said, I am currently Director of Bioinformatics at AstraZeneca, and we support all the data analysis needs from preclinical scientists. So mainly focus on sequencing data, bulk RNA sequencing, DNA sequencing, and a lot of also epigenomic profiling data as well, like such as ATAC-seq, cut and run, and also DNA methylation data. So we are supporting the preclinical studies from new target identification, MOA studies, and also maybe preclinical biomarker identification.
Awesome. Fantastic. So with that being said, Tommy, I want to start with talking about your journey first. You've had a remarkable journey, right? Building a distinguished career in computational biology across Dana-Farber, Harvard, and MD Anderson. And now you're AstraZeneca, all while building a professional network, navigating the new environment. So looking back, what would you say was the pivotal moment where you decided that this is going to be my trajectory?
So there are a couple of different pivotal moments. I can talk about the one that after I came to the States. So I'm from China originally, came to the States 2008, August 8th. That's a very easy to remember date. So I came to University of Florida to pursue my PhD in molecular cancer biology wet lab. And I was trained in a molecular cancer biology wet lab, and I was doing a lot of pipetting, all those biochemical studies, experiments like Western blot. I even did some Northern blot in the old days. So I was doing really well actually. In the fourth year of my PhD, I already published first all the papers, and I was feeling really good because I can graduate now because with 2 publications. Then my PhD advisor asked me to analyze this public available chip sequencing data. So it's a sequencing technology to identify global binding sites of transcriptional factors. And I downloaded data, it was 2 gigabytes, and I just couldn't analyze it because it crashed my Excel. So that's the pivotal moment that I realized I didn't have any data analysis skills. So I started to learn by myself. So I took a lot of courses, like almost 40 courses in the first couple of years on those platforms. Coursera was just studying, later edX, Udacity. I really grateful for those platforms that I can actually have the opportunity to learn essentially statistics, programming languages from those top niche actually universities. So I think that probably was the pivotal moment. And then since then, it's been 14 years, I think, since I typed first Hello World. And now I'm just doing full computational biology work. And as you mentioned that I'm passionate about education, so that's why I share all the tips, tutorials in my blog and also on my LinkedIn, essentially just teach people how to do bioinformatics, because I think biology has become a data-intensive subject, and we all need at least some data analysis skills to be able to deal with data.
Got it. So for listeners who are not in your world, Tommy, what would you say a computational biologist, what does a computational biologist do, and why does it matter?
Yeah, computational biology or bioinformatics they're very big, it's a big field, and there are a lot of things people doing. And I'm using actually bioinformatics and computational biology, these 2 terms interchangeably. People think there are differences, but I just feel it's not that helpful to distinguish these two, because someone's bioinformatics is someone else computational biology. But in essence, we deal with data that generated from the biological systems. And for me, I deal with a lot of sequencing data, essentially next generation sequencing. So you can sequence RNA. So it's called RNA sequencing, probably is one of the most commonly used sequencing technology to look at gene expression profile across all the genes in a sample. Or now we have single cell RNA sequencing, so you can look at gene expression level at a single cell resolution. So my job essentially, so the wet lab people, they will do the experiments, perform the sequencing experiments, and then we took it over, try to analyze data, and derive biological insights and try to inform the decisions for drug development. For example, like in the pharma setting, we can perform experiments. For example, on cell lines, you treat the cell lines with a couple different drugs or different probes for your drug. So you're different, because before lead optimization, you might have a couple of different candidates. Then you can perform RNA sequencing experiments and look at the gene expression changes for different, then compare their profiles across different compounds and see, okay, maybe a certain compound is not specific enough, it's hitting some off targets. So you can actually identify those off-target effects by studying the gene expression profile and also pathway analysis.
Okay, amazing. Thank you for letting us know that. Now I want to switch gears a little bit. I want to talk about the part around the preparation, right? How do you get your data ready for artificial intelligence or any kind of analysis? And so one of my first question is, when you receive the data, what part of it is actually analysis and what part is really cleaning the data, scrubbing the data to make sure you can actually even use it?
Yeah, so I know AI is kind of very hot at this moment. But before AI can make a big impact, you need to make the data AI ready or make it tidy, right? I think the first step is always actually tidying the data. Like my own experience is that even the biologists, they perform the experiments and there's still a lot of actually data cleaning need to be done. For example, the same samples or even the same cell lines, they may perform RNA sequencing and also DNA sequencing on the same samples. But because there's like no good, routine they follow, okay, they actually name those samples kind of differently. And as a data scientist, when you get the data, you have to actually match all those different samples because they have maybe little bit different sample names. Okay, hyphen versus underscore, capital versus lowercase, for example. So you kind of have to actually clean those data first. I think within the company, it's probably less of a problem, but if you're dealing with public available data, the metadata, which is the data describing the data, is very messy as well. You have to actually harmonize them because, for example, like the same column, they mean the same thing, but they use slightly different column names. So you have to actually harmonize them. But then after you get the data, you have to do actually heavy quality control QC of the data to make sure it's of good quality. So before you do any real analysis, you want to make sure the data is good, because data is not just rows and columns when data scientists see them. Before you see the columns and rows, there's a lot of things going on, right? The cells, maybe you culture them in a not optimal condition and the cells don't behave well, and then you extract DNA or RNA, there could be something wrong with the data as well. And the other thing is that it's very easy to make mistakes as well. Like people can swap sample labels, because when you have a lot of samples, it's just very easy to make mistakes. And you want to do this quality control, like principal component analysis, and to find, okay, maybe I have some sample swap. For example, you have knockdown, you have drug treatment versus control, then you have replicates or triplicates for each condition. And if you do a PCA, you should see, okay, the control samples, they form a group, and then the treatment samples, they form another group. But if you see something, you have 2 points here, that's the control, but then you have another point that is close to them that is treatment, there could be some sample swap. So you need to identify those some mislabeled samples before you carry out the downstream analysis.
Right, right. No, very well said. And I will, I'll also relate this to my world in manufacturing and supply chain, where a lot of times when you have the data, it's not clean. Sometimes it's scattered all over the place. So you really have to do a lot of activities to make sure that it is clean. And it is interesting to see that that problem is, or that issue is prevalent all throughout all the way from the beginning of the product life cycle towards the end. So let me ask you this: what has been your experience with initiatives where the data was not clean, where there were issues in the data? What impact has that particular issue had on a project compared to when you were able to clean the data completely?
Yeah, so I think there's a difference between actually small biotech and also big pharma companies, because big pharma companies, they already have their systems established and they have actually been using this old system for a long time. So it's kind of hard for them to make changes, especially for the old data. I know there's a gold mine actually in all this old data, but cleaning all this old data require a lot of efforts and resource.
Right.
And then it's a matter of if the project is already closed, who is going to, like, what's the value actually to go back and clean the data again, right? But then when we look forward, maybe we can still maybe enforce this, okay, new data standard or data model. Okay, let's input all the same actually labels for the same variables for each experiment. So there's a database behind that so people can easily find them, right? I think for maybe clinical trial data, like there's a little bit more rigid way for metadata management, but for preclinical data, I think there are a lot of more actually flexible spaces because the experiments are very different for different projects, and there could be a lot of actually free labels from the scientists that is not actually compatible across different projects. Somehow manually map different metadata. That actually takes a lot of time. And then in a small biotech, if you are just studying, and you might, let's just follow the best practice in day one. Okay, let's enforce all these experimental biologists to input the metadata in a consistent manner, and then those actually are linked into the whatever digital notebook and maybe tools like Benchling. So the data scientists can easily pull out those data and do metadata analysis. I think if we do have that setup, it can save us a lot of time.
I see. Okay. That is very interesting. And that's good to know as well. What's the biggest gap you've seen as well? Anything else besides you already mentioned in terms of the organization starting to get ready to deploy AI or automation?
What's the biggest gap? Again, it's data, right? I think AI needs a lot of data to train, right? Like, it really depends on how you define AI. AI, artificial intelligence, is a very big term. But the most valuable thing that AI can deliver, like, for drug development, of course, I can say it's in the clinical trial, space, because clinical trial usually is the bottleneck of the whole process.
Right.
And of course, AI can help to speed up the R&D, the research and development as well. For example, like you can train an AI system to improve the property of those small molecules, like make sure they are more soluble, and maybe they have more specificity. And even if you have a big compound library, you have maybe millions of them, and you can do a virtual screening using those AI docking systems, and to reduce the searching space to a smaller amount, and then you carry out the experiment. So in a sense, you can save time and money. Yeah, but the gap still is the data. I think for clinical trial, how do you actually recruit the right patients using the right biomarker? That's a $1 billion question, because each drug costs you like $3 to $5 billion to make. If you can crack this problem and you recruit the right patients to increase the successful rate of clinical trials, that's going to be a big impact. But the problem again is that the data is messy as well, because different patients, they have very heterogeneous treating, for example, history, and they have different genomic profile, they have different, maybe genetic background. All those can actually add noise actually to your AI or machine learning algorithms. And you have to be really careful whether your AI algorithm is picking up those noises or finding true biological signals. And again, the data is not that huge, right? I don't know, for phase 1 clinical trial, you have like 40, 60 patients, right? Phase 2, you can have a couple hundred. Like phase 3, maybe you can get to 1,000.
Right.
But still, like in terms of sample size, like in terms of patient samples, like that's only within thousands, right? That's still a small number, of course. Biological data, depending on how you look at, like if you do single cell experiments, a single sample can have millions of cells, then you think it's a big data. But again, in terms of actually number of patients, it's still within thousands. So that's not a lot of number for AI, like, in my opinion.
Right. Yeah. So that is a big challenge where there's not enough data to that's interesting. So how is that problem addressed in your perspective?
I think people are trying to address it by combining different cohorts together, right? Like for
I
a
see.
single trial, maybe you only have hundreds and thousands of samples. But then if you combine all different results to data from different trials together, or maybe there's some actual data vendors such as Tempus, like Caris Life Science, they actually are accumulating those genomic data sets from real patients. I think they have some sourcing from the hospitals. So it can get into like hundreds of thousands. Maybe you can still do some machine learning stuff with those data. But again, heterogeneous patients can give you some troubles, and also the genomic background is different. Like for example, if you want to look at specifically KRAS mutants, KRAS mutated patients, then you suddenly reduce the number of patients to a subset. So for example.
Yeah, yeah, no, no, very good input, and thank you for mentioning that. Even in my own experience, working in life sciences, I've seen that firsthand. How long it takes for a drug to start from the beginning, right, phase 1, all the way to phase 3, and then post approval. So it's a long cycle, and I think anything we can do to further make it more comfortable or automate it by to your point, by getting the right data, cleaning that data can be very helpful.
Right.
So yeah, no, that sounds good. Now I want to switch gears to your content creator angle, right? You've done an amazing job building up your followership, writing so many blogs. What has drove you to do that? I'm curious.
Drives me. So I've been writing blog posts for over 10 years. I mean, since I started to learn bioinformatics from scratch. I mean, to be honest, initially I was a little bit selfish. I just wrote it down because I encountered this problem that I didn't know how to solve it. I was just trying to take notes, and then I wrote blog posts. And gradually people probably Googled, and because they had the same problem, then they landed on my blog post. I think initially it was just for my own, interest, because I always need to go back to check my notes, and if I have a blog, then I can always easily find them. Yeah, that's what initially drives me, but then later, like a lot of people actually recognize my name, my blog in the conferences, and then I try to be a little bit more educative. So, I find actually teaching is very interesting. I like teaching, and try to explain all those complicated concepts in a little bit simpler method. And also I went from this transition from a wet lab scientist to a computational scientist, and kind of understand the mental barrier in the transition. So I want to share some of my tips and try to shortcut other people's learning process. And that's for blog. And for LinkedIn posts, I've been posting actually for almost 780 days, actually every day consistently.
Amazing, amazing.
Yeah. So I think there's no secret. If there's a secret, I will tell you, you just need to be consistent. And if you show up every day, even if you don't feel in the right mood, you will get the results. Yeah.
I love it. I love it. And for any of our listeners who are looking to build followership who are really looking to set a brand for themselves, I think Tommy presents a great example of how he has done it over the years. As I was reading more about your profile, one of the things that stood out to me was your motto, which says curing diseases with data. Can
Right.
you talk a little bit more about that? What does that mean to you? And how do you see that impacting people around you, the world around you?
Yeah, so you may notice that I didn't say, okay, cure disease with AI because, again, we talked about it. AI need the oil is the data. So we really need to create a system to generate data that can be directly feed into the AI system. I think that's the first step. Before you have any AI strategy, you
Yeah.
need to have a data strategy. That's been always like my motto because I think that different ways to look at it, like data, if you're doing actually high throughput, for example, experiments, actually you can design an automated lab system and then generate those data in a very consistent manner, right? And then you also have the metadata traction actually built in the system. So I think for those high throughput screening, for example, high throughput imaging analysis, for example, it's actually very beneficial to build the system and also essentially store all the data in a consistent manner, and then that directly feed into the algorithm, and the algorithm maybe will give you some feedback to change any conditions. And I see this flywheel going on in the lab and also in the computer. So that's why I think we can cure disease with data, but of course by the advancement of those AI algorithms as well.
Tommy, I also believe that very strongly that you have to have the right foundations to deploy any kind of AI, which includes data foundation, process foundation, technical infrastructure, organizational effectiveness. So these are some really, really important factors. Now, let me ask you this, right? You've shared a lot of content online, it's all free. What and I can see that you're very, very transparent about it. So what has that opened any doors for you? Has that done something to progress your career or your own personal knowledge versus if you had not done that?
Yeah, so the first benefit I will say I actually get to learn the things better myself because if I need to write it down and to teach other people, I need to understand it better myself. So I would
Right.
say that's the first benefits. And also it's a long-term effect. So you have to actually do this actually for a long time. I think once I heard sentence is okay, you need to pick a simple thing, take it serious and do it for a long time. Like my time horizon is like always like 10 years. So picture yourself who you want to be in 10 years and you then just work toward that, right? And for me, because I have been writing blogs for over 10 it does open actually many new opportunities for me because people got to know me before even I went to the interview. Maybe some of the interviewers, they already knew me through my blogs and they know actually Tommy is capable of doing that. He's an expert in sequencing data analysis. Definitely it helped me a lot. I would encourage everybody to start building your online presence. At least have your website made because when people search Google, ChatGPT or Claude, if they can't find you, like you don't really exist because people like you try to Google you, or maybe if you don't have those websites, at least you should have a really polished LinkedIn profile.
Yeah, that is such an important point. I've seen students where they're really struggling to land jobs if they all they have done is focused on studies. So I highly encourage everybody to truly get outside, get a different experience because that will make a difference. So now let me ask you this, Tommy. Looking back in your career, what is one thing or what are maybe two things that you wish you had known at the beginning of your career? Now that you know now.
One thing that I think the most important thing I would say is probably build your like brand, personal brand. I had no idea what was a brand, and since I started maybe 10, 14 years ago, I attended this online actually leadership boot camp, and the coach talked about the personal branding the first time actually I hear about it. So that was in 2020. So actually I started not too actually long ago, like 5, 6 years ago. But once you actually hear about that, Now you cannot unsee it, so you need to take action. So I think if you start it right now, you have a really good starting point. So I always tell people, okay, the best time to start was 10 years ago, but the second best time is always right now. So it's never too late. That you have to be persistent, and because sometimes people post, okay, for a month and say, oh, it doesn't work, but you just really need to do it at least for example, 1,000 days. And if you have that commitment, and I'm sure you will see the results.
Yeah, absolutely, absolutely. And this podcast is about having honest conversations around AI technology and leadership, right? As we talked about. So let me ask you one of the last questions from my end. What is one thing that you wish your leaders or leaders in general knew about deploying automation? They may not be that they may not be well aware of or may not be paying too much attention to.
How the workflow is done. So when they are trying to implement AI strategies, they really need to get some feedback from the people who are doing the real work. So getting some feedback, okay, this step maybe can be automated by AI, and that step maybe not, right? So they need to have a very clear picture of how the bottom work, how that was done. I think getting some familiarity of how the day-to-day work is done and getting feedback from the scientists who are performing the experiments, who are analyzing the data, can have a huge impact on how they really implement those AI strategies.
100%, 100%. I think the more we work together with a team who lives this process day in and day out, the better we are off, right? So I fully agree with that sentiment. So with that, thank you very, very much. Really appreciate your time. Is there any closing comments you have for our listeners? Anything you would like to leave them with?
AI is actually advancing so fast, and when we talk about AI, we are mainly referring to the LLM, like large language models like ChatGPT or Claude. So I will encourage everybody to embrace them. Try to learn all those new technologies and the application side, and just play with it, right? So if you have a repetitive work during your like everyday work, and try to automate some of them, use those new tools. And for me, like as a bioinformatician, I'm actually using the tools such as Claude Code to write a lot of code for me, right? It's much faster. But again, you need to know what questions to ask. You need to be able to judge the output of the AI. Sometimes AI give you, the code that runs fine, it just gives you the wrong answers. Still takes years of experience to judge, and also to instruct AI to write the right code for you.
Yeah, absolutely, absolutely. And I fully agree with the fact that you really have to continue to learn. In fact, that's the reason why at SkyPreme as well, we run boot camps live and in person as well as online. So highly encourage in general for everyone to learn about AI in their daily life. So with that being said, how do
So they can find me on LinkedIn. So you just search Tommy Tang. You should find me on my LinkedIn, and then you can find everything else
Awesome.
from there.
Awesome.
Yeah.
Awesome. Thank you. Thank you, Tommy. And for our listeners, thanks again for listening in. If this interests you, please look forward to the next episode and visit skypreme.com. So with that being said, thank you so much for everything. And we'll see you next time.